We are a group of undergraduate students from IISER Thiruvananthapuram who decided to leverage the powers of synthetic biology to tackle the issues of fungal infestation using the iGEM platform. Firstly, I'd like to talk about how it all began. Taking inspiration from various projects, we explored many different issues, primarily prioritizing environmental concerns. We observed black patches on walls, which we discovered are caused due to fungi that are not only nasty to look at, but are also a reason for a plethora of respiratory illnesses. We later collected samples from our campus and subcultured them to identify the species using Sanger sequencing. We successfully identified 7 species of fungi, which included Aspergillus niger, Aspergillus versicolor, and Rhizopus oryzae, to name a few. We decided to engineer recombinant chitinases with the aim of developing a novel, chimeric antifungal enzyme to eradicate black mold thriving on the walls of buildings. Chitinases are enzymes with the ability to degrade chitin polymers, compromising fungal cell integrity and ultimately leading to their death. Most wild-type chitinases have a chitin insertion domain or CID and a catalytic domain. Our chitinase design involves joining domains from different wild-type chitinases, leading to the formation of a hybrid chitinase. In order to rationalize the design of our chimeric chitinases, we consulted with 2 renowned crystallographers, Professor M.R.N. Murthy and Professor H.S. Savitri from ISC Bangalore. We were faced with the unique issue that those chitinases, which were effective against diverse fungal species, such as Serratia martians, failed to retain significant fraction of their activity at physiological temperature and pH, which is critical for the success of our experiments. The experts suggested that it is the catalytic domain of chitinases which determine their temperature and pH characteristics, and hence we chose a favorable catalytic domain from Pseudoaltermonas to join with Serratia. The experts also suggested that we use chitinases from plants for 2 reasons. Number 1, they display activity against a range of fungal species with optimum temperature and pH in the required range, and number 2, we assume that the chimeric chitinase won't cause any health issues since they are edible to human beings. Based on this, we designed 2 plant chitinase combinations, PC1 and PC2, joining domains from barley and wheat respectively. Dr. Binod Parameswaran is an expert with several years of experience in the field of designing chitinases for applications in both healthcare and agriculture. He pointed out the difficulties of manufacturing an enzyme for application in external settings due to concerns over its stability and the high cost to benefit ratio. He suggested that we could make a more significant impact to our project by showing us the plight caused by fungi in mainly 2 domains, health and agriculture. In order to further investigate in this direction, we engaged in discussion with several stakeholders such as doctors and farmers who confirmed to us that the burden of fungal infestation and contamination in both healthcare and agriculture is certainly very large. We came to appreciate that fungal spores cause a plethora of diseases and that the unregulated use of fungicides in agriculture has severe negative implications for the environment. Additionally, fungal infections cause around 1.7 million deaths annually, highlighting the global nature of the problem. In addition, current antifungal treatments suffer from the issue of development of drug-resistant fungal strains and may also cause additional complications such as nephrotoxicity. Consequently, we were open to 2 choices. Number 1, an antifungal solution to the havoc wreaked by losses in agriculture due to fungal infestation, and number 2, an antifungal therapeutic solution to fungal infections in humans. During this time, India witnessed an enormous soar in the number of mucormycosis cases as a post-COVID complication. This essentially helped us in resolving the dilemma, and we chose to work on the former option, leading to the conceptualization of Moldomort, a class of antifungal therapeutics to invasive fungal infections. In addition to the recombinant genes we had already designed, our team developed another chimeric bacterial chitinase, mainly to target Rhizopus species, which is one of the main causative agents of mucormycosis. We chose A. orientalis due to its efficiency in lysing the chitin walls of Rhizopus and mucor species, which are the major sources of mucormycosis. In order to increase its efficiency, we added an extra chitin-binding domain from Serratia to A. orientalis chitinase since its CBD is not yet annotated. We call this combo BC2. Our wet lab work was majorly divided into 4 parts: cloning, expression and purification, enzyme characterization, and antifungal assays. We successfully cloned and expressed one of our hybrid chitinases, Bacterial combo 2, and a corresponding control, Streptomyces wild-type chitinase in E. coli BL21 strain. We carried out IPTG induction and expression in different sets of IPTG concentrations and temperatures and decided to purify protein through nickel NTA resin. However, the protein was eluted in the flow-through and not binding to the column. In order to expose the His tag, we treated protein with urea, which helps in denaturing the protein. This is followed by stepwise dialysis. During the last phase of dialysis, we observed suspended precipitates in the dialysis bag which had formed due to the aggregation of our protein. Thus, instead of dialysis, we did ethanol precipitation and dissolved the protein in a buffer containing 1x PBS, followed by concentration through an Amicon filter. In this way, we purified our protein. Next, we tried to characterize our enzyme, and for this, we employed both the DNS assay and circular dichroism spectroscopy. Initially, the activity of our enzyme was calculated using the DNS assay, which is a colorimetric assay. Upon incubation of our chitinase, which is a substrate, which is colloidal chitin, the enzyme hydrolyzes the substrate to produce a reducing sugar, N-acetyl glucosamine, which can be quantified with the help of DNS reagent. With the help of a standard graph, the activity of bacterial combo 2 chitinase at 40 degrees Celsius was found to be 0.56 units per milligram. Additionally, we noticed that beta sheets were relatively stable at higher temperatures than the alpha helix, which had a melting temperature of around 92.58 degrees Celsius. Furthermore, we characterized the integrity of the secondary structures with an increase in temperature starting at 20 degrees Celsius to 100 degrees Celsius in intervals of 5 degrees Celsius using circular dichroism spectroscopy. We observed that the plotted curve for our protein started to significantly deviate from the initial curve at higher temperatures. Our antifungal assays involved determining the zone of inhibition when our chimeric chitinases was spotted on pure fungal cultures. Firstly, we tried to determine the zone of inhibition by bacterial chitinase combo 2 or BC2 against various fungal species, and we observed significant chitinolytic activity at concentrations greater than 1 mg per ml. Next, we compared the zone of inhibition by BC2 and Streptomyces chitinase, otherwise known as chitinase from A. orientalis, on Rhizopus oryzae. The results were inconclusive, and a potential reason for this could be that BC2 was purified several days before Streptomyces, and hence its activity could have slightly degraded over time. Coming to project safety, we started off by getting approval from our Institute Biosafety Committee or IBSC. Secondly, we ensured safe environment interaction, meaning that our protein of interest is not secreted outside the laboratory, preventing its release in the environment. Additionally, we used only monitored risk group organisms involving fungi belonging to only risk groups 1 and 2. No organism with risk group higher than 2 was used in the study. Also, we ensured to follow proper laboratory safety protocols and waste segregation to ensure safety of the project.
The focus of the dry lab at iGEM IISER Thiruvananthapuram was to develop, build, and test models to predict and verify the experimental results in a mathematical formulation. Through modeling, we were able to predict the results of various experiments performed in the lab and other experiments which were not practically possible under limited time and resources, which also solved the optimization problem of the wet lab members. During the initial phase of our project, we set out some goals predicting the efficiency of a recombinant enzyme through various tools such as Swiss model, RaptorX, and Autodoc Vina, developing and testing fungal growth models, determining the decay efficiency of our novel antifungal, predicting activity of a recombinant enzyme, estimating and optimizing protein output upon induction of IPTG, and formulating mathematical models to support our claim to use nanoencapsulation as a delivery mechanism. Let's start with structure modeling. The recombinant proteins are synthetically designed, hence its efficiency to bind to ligand is not known through any literature. Therefore, to understand its binding efficiency, we conducted a docking process with chitin octamer. Usually, -6 kilocalorie per mole indicates a good binding between ligand and receptor. We have found around -10 to -12 kilocalorie per mole for a recombinant enzyme. This definitely has backed our hypothesis of creating antifungal with good binding efficiency. Moreover, using a set of ordinary differential equation and Autodoc data, we determine ligand receptor complex concentration at any given time for both wild type and recombinant chitinases. This proved that adding binding domains from a high efficiency enzyme, here Serratia marcins chitinase, to catalytic domain of low efficiency enzyme, that is Streptomyces orientalis, indeed increases the binding efficiency of low efficiency chitinase, and thus the recombinant chitinase bacterial combo 2 has higher binding efficiency than its wild type counterpart Streptomyces orientalis. Moving on to fungal growth models. Growth models in general serve the essential purpose of providing us with the population dynamics of a species. Models often predict parameters such as growth rate and lag time of a species, thus providing us with an analytical tool to study the promotion or demotion of the population as a result of change in external factors such as temperature, pH, chemical, or as in our case, antifungal. For our project, we used two fungal species, Aspergillus versicolor and Rhizopus oryzae. We tried out various experimental methods to measure the growth rate of fungus as a function of time, and we finally settled on two of them, surface area based growth models and optical density based growth models. These methods worked pretty well for our expectation and gave us the well-known sigmoid shaped curve for growth, demonstrating lag, exponential, and stationary phases. We tried to fit our data with well-known growth models such as Gompertz, Logistic, and Richards to determine the growth rate and lag time. Gompertz gave us the best result with our experimental data, that is lowest RMSE and highest R-square values. Our next model focuses on determining IC50 of drug against these fungal species as a measure of degradation potency of drug. IC50 is defined as the concentration of drug required to achieve inhibition of growth by 50%. It is a widely used tool to measure the degradation efficiency of a drug. We planned to determine the IC50 of our protein but were hit by a major setback due to lack of time. Overcoming this setback, we decided to use commonly known antifungal, Nipagin as proxy molecule and proceeded with the experiments, we were able to determine and reproduce the results of our IC50 experiments, which will act as a testbed for comparison with IC50 of our novel chitinase in future. To predict the activity of our novel chitinase, bacterial combo 2, we used a modified version of Michaelis-Menten equation which considers the addition of binding domain to the catalytic domain of chitinase. Through autodoc data and an extensive literature survey, we were able to determine the necessary parameters which help us to plot the kinetics curve. Through Michaelis-Menten's curve and its associative Lineweaver-Burke plot, we were able to predict Vmax and Km for a recombinant enzyme. Since our gene is under inducible promoter, determining the optimum amount of inducer molecule, IPTG here can predict the maximum output of protein. The T7 polymerase gene and our gene of interest are both under lac-operon. To model the protein output as a function of IPTG induced, we tried to model every cellular process involved in induction process from scratch through ordinary differential equation and successfully built a model for IPTG optimization. For this, we collaborated with IGM Ecuador 2021. This model supported the wet lab, providing the optimum amount of IPTG that should be used to maximize the production of our enzyme. Further, the experimental protein output showed an excellent match with our prediction.
For any drug, identifying the right amount of drug and the exact time it requires to achieve the desired result should be determined. In case of fungal infection, both causative organism as well as the target site are diverse. After getting approval from our institute, we initially conducted a survey to learn more about the drug preference in the general public. The results showed that the people prefer medicine in the form of chemicals rather than GMO, and the majority preferred encapsulated drugs. Upon interaction with Dr. Rachit Agarwal, we decided to use polylacticoglycolic acid based nanoparticles due to their versatility and modularity. Adjustments in the polymer molecular weight, lactate glycolide ratio, and the concentration of the drug will enable release of desired drug dosage. In order to facilitate active targeting of our nanoparticles, we intend to tag them with dectin receptors, which will bind to beta-glucans present in the fungal cell wall. Upon binding, the nanoparticles will be degraded through ester bond hydrolysis to release our enzyme. Since degradation of PLGA is directly proportional to release of our enzyme, we could estimate the amount of drug release as a function of time. We predicted that it will approximately take 40 to 50 days for its complete degradation, which also supports the idea of a controlled and prolonged release of a drug. We knew that if we want to develop this drug further, we would have to perform several preclinical trials. For this, we sought help from Dr. Gui, an expert in pharmaceutical industry. He helped us in overall view of the tests and phases through which our drug would have to be assessed, one of which included assessing the cytotoxic activity of our chitinase in various cellular models and organoids. As an extension to our project, we aim to include glucanase along with our drug to act on glucans present on fungal cell walls. Since the solution now targets multiple components, it has an added advantage of decreased antifungal resistance and increased activity. This idea was also backed up by our interaction with Dr. Robin Alshire from University of Edinburgh. Our chimeric chitinase can be effectively used as potential biocontrol agents in agriculture as well. As our project largely deals with chimeric chitinases, we aim to employ the same logic to construct human chimeric chitinase. We hypothesized that such a chitinase would have a much lower immune response and could work as an efficient therapeutic. Another arena where we aim to employ our chimeric chitinase are in the removal of walled molds that can help to reduce the extent of fungal infections. Upon interacting with Dr. Ravi Shankar, an expert in veterinary sciences, we came to know that animals usually face tropical fungal infections against which a cheap antifungal ointment would be useful.
Next, we moved to our human practices and outreach section. Throughout the project, we met several experts and stakeholders such as structural biologists, industrial experts, social scientists, law experts, medical experts, and farmers. Their suggestions helped us improve our project design, wet lab experiments, outline a proposal for implementation, design surveys, and improvise our handbook on the in-depth legal scenario of biotechnology. Identifying potential stakeholders of the therapeutic we were developing was crucial to the advancement of the project. Patients, medical doctors, caretakers of patients, NGOs, biopharma companies, and the general public are our major stakeholders. The initial paradigm shift from the environment track to therapeutic was also a result of our interaction with the experts and stakeholders. Due to several limitations set by COVID-19, we opted for a literature survey to understand the incidence and severity of fungal infections, particularly in immunocompromised patients. Fungal infections are a major cause of concern to immunocompromised individuals like AIDS patients and can lead to fatality if not diagnosed and treated early. While interacting with Father Jijo, We learned that AIDS patients regularly visit antiretroviral therapy or ART centers. Hence, distributing our antifungal drug through these centers would make them more accessible to the target users. We also had a meeting with Dr. David Denning of Gafi, an NGO working to raise awareness and collect worldwide data on fungal diseases. He encouraged us to build a target product profile for our drug and look into the allergic responses. After we settled on therapeutic to be our project's primary goal, we needed to learn the awareness of fungi among people. We surveyed the general public and surprisingly understood that people were generally unaware of fungi and fungal infection. To address this, we began by dividing the general public into 3 categories based on age: kids, teenagers, and adults. For children, we designed a book named The World of Fungi with illustrations and short notes about the various types of fungi. For children between the ages of 11 and 16, we conducted Dear Fungi, letter writing competition on the occasion of our National Reading Day, where participants were required to write a letter to a fungus of their choice after reading about them. For adults, we organized The Dark Side of Fungi to generate awareness among pathogenic fungi. The event hosted talks on infectious diseases and antifungal drug resistance along with our project presentation. Education and communication. All activities we conducted as part of education and outreach was carefully designed to cater to the different levels of understanding about synthetic biology in different age groups. SynSync, a synthetic biology book for middle schoolers. The book provides a brief introduction to synthetic biology while explaining the fundamentals of recombinant DNA technology. The book's highlights include simplified concepts, illustrations, did you know boxes, and games. SynBio 1.0 was an entirely cost-free 4-day-long synthetic biology course workshop conducted for 11th and 12th graders of Rajagiri Public School. The first 3 days dealt with the basics of synthetic biology, while the 4th day was dedicated to our project presentation. We shared the presentation study material with the school for future reference. Splice and Connect were intercollegiate synthetic biology themed games that enticed massive participation from colleges all over India. Introducing iGEM to the juniors helped introduce different concepts and techniques of synthetic biology. As a result of the presentation, many of them came up with interesting project ideas as well to participate in the next iGEM competition. A talk series highlighting the vast applications of synthetic biology in various fields seemed an engaging way to unveil synthetic biology, a developing area of science. It hosted 5 different speakers from different walks of synthetic biology. Participants were highly enthralled by the application of synthetic biology in various fields of science. Considering the relevance of the project in the COVID scenario, the impact our project can have on society attracted media attention. This served as a tool to communicate our project with the masses. Our project was in the news outlet The New India Express and India Bioscience. Exhibit A, the official publication of the Science and Technology Council of IISER Thiruvananthapuram, approached us for an interview where we talked about the conception of our project idea, our inspiration, and the changes we made to it. This year, our team, in collaboration with Team iGEM IISER Tirupati, prepared a handbook summarizing a few Indian biotechnology laws aimed at helping novice researchers in India. This was done by incorporating suggestions of law experts. Furthermore, we hope that it will serve as a guide for upcoming Indian iGEM teams. Inclusivity. Recognizing the importance of including diverse sections of society in science, our team conducted several activities to facilitate inclusivity in science. Sky Onam was an attempt to bridge the gap between science and art by incorporating scientific designs into traditional floral carpets made during Onam, Harvest Festival of Kerala. Bio Explorer was another initiative by the team to educate students at niche with hearing impairments. We created a presentation explaining the basics of synthetic biology along with a supplementary description document to help the interpreters. Our next initiative was to study economic inclusivity in iGEM. Using Human Development Index as a parameter, we arrived at the conclusion that participation in iGEM from both low and medium HDI will only add up to 5%, while the remaining 95% is from countries with high HDI. Furthermore, we also suggested remedial measures to dissolve this disparity caused due to economic status. We started a hashtag challenge on Instagram, SynBio on Food, which was an attempt to spread synthetic biology by designing scientific entities on food. Throughout iGEM, our team has successfully collaborated with various other teams from all around the world. Here, we'll present to you the team behind the iGEM project. We thank our PIs for their selfless support in making our dream project a reality. We also thank our mentor, advisors, and instructor for guiding us in every step. Finally, we thank all our sponsors who believed and stood by us. Thank you.